A lot of familiar faces and friends here, and new faces as well. This is exciting because we are all about friendship in Racing is the Clock, and I'm sure in ValleyDAO as well. So, yeah.
Definitely. The journal club was, I would say, heavily oversubscribed. So there's been a great interest in being part of this. So hopefully we can keep up the momentum and see it to grow into, having more researchers coming in and guesting, talk about their research, and also at some point having the community recommend research articles that we can discuss in future meetings like this.
Yeah, this is going to be fun. So since we have a fully agenda packed, let me get started. So welcome everyone to the Racing is the Clock and ValleyDAO Journal Club. In this first session, we will discuss the paper Machine Learning Aid Engineering of Hydrolases for PET depolymerization. I hope I say it well. Basically, in simple words, badass enzymes to break down plastic. So no one gets plastic, like we need those enzymes. So many of you know the purpose of the Racing is the Clock. We are in a race to engineer biology to fix the planet and avoid extinction. So we are a community of aspiring and early stage biofounders and agents of change determined to energize the bioeconomy. As part of Racing is the Clock, we host a yearly summit in the SynBioBeta Build with Biology conference. And through the year, we organize virtual workshops and events like these, and we're planning as well to do some regional meetups. But the most important here at Racing is the Clock is that we are about all friendship. Like, our culture is to be kind, our culture is to be collaborative. We really need to help each other if we want to create a flourishing bioeconomy. So if you join our community, you will expect a very friendly and informal atmosphere. So now I will pass the mic to my friend, Albert Anis, who is going to introduce what's ValleyDAO, the general purpose of this journal club, and he's going to introduce our special guest of today. So Albert, all yours.
Yeah, thanks a lot for that, Ivan. Let me just share my screen. Hopefully everyone can see. Yes. So yeah, hi everyone. My name is Albert Anis, and I'm one of the founders of ValleyDAO. And like many of you, I'm also a former iGEMer and advocate of synthetic biology as a very detrimental tool in the transition to a sustainable bioeconomy. I proposed the idea of initiating a journal club in the Race Against the Clock channel, and seeing the immense interest, I thought it would be nice to also invite along the ValleyDAO community. And so our intention with this journal club really is to highlight exceptional and interesting synbio research that directly tackles the problem of climate change. So that will become kind of the theme of the journal club. And also, of course, provide a chance for the synbio community to come together, discuss, and directly interact and ask questions to the authors. So also many of you might be wondering what ValleyDAO is. So I will go over that briefly. So at ValleyDAO, we believe that biomanufacturing has the potential to drastically improve human and planetary health. However, with any breakthrough technology, such as pharmacology or the internet, there will always be some form of incentives for misuse or monopolization or corruption in some form. And we believe that synthetic biology won't be an exception for this phenomenon. And we can already see kind of traces and hints of this. So I call it the synbio IP wars. One example is we have Impossible Foods suing motif for using a similar protein to what they are using in their meat, but it's not exactly the same. And what this does is more or less, it not only is it stifling competition, it is also stifling innovation. And in order to really achieve the ambitions of a sustainable bioeconomy, it is going to dependent on access to these technologies. And we believe it all boils down to the ownership of intellectual property and how that is managed. So what we really need is a more democratized way of deciding how IP in synthetic biology is licensed and distributed. And that's exactly what we're trying to solve at ValleyDAO. So ValleyDAO, you can see it as a community that funds and acts in the best interest of the synth bio ecosystem. And a way to ensure that it acts in the best interest of the ecosystem is by allowing the community members to themselves decide how intellectual property is distributed. So at ValleyDAO, the community will be able to recommend and vouch for different academic research projects. And then the members of ValleyDAO, so the community members, which can be anyone, will work in reviewing the research and finally cast their votes to decide if the researcher get the funding or not. And on average, we'll be paying out approximately $100,000 to $150,000 grants under the agreement that any generated IP from that research projects goes back into the community. And then it is up to the community to decide how that IP is used. So one thing that the community can decide is okay, let's license this IP to this company working in producing a certain biomaterial. And what this ensures is that that company doesn't have a monopoly on that IP, but ValleyDAO can also sub-license that IP to other companies as well. What ValleyDAO could do also is to spin out this IP into startups, and using ValleyDAO's network to find talent, to put together a team, to do the fundraising, and so on. So that's a bit of ValleyDAO. There's of course a much more complexity to this. I didn't really go into the technology that will be applied to facilitate community voting and IP transfer and so on. But if you go here to our link tree, and we will send out this later through a follow-up email, you can read more about how we're tapping into Web3 to facilitate kind of these community activities. So that was on ValleyDAO, but I would like to now hand over the word to our visiting guest, Daniel J. Diaz from University of Texas of Austin, and he will give a brief 10-12 minute presentation on the research, and maybe also give us some anecdotes and some exciting stories that have gone into this work. So I will stop sharing.
Hello, can everybody hear me?
Yeah, we can hear you well, Daniel.
Yes.
All good.
Perfect. So I want to appreciate you guys for inviting me to talk about our recent publication. It was pretty exciting to share it with the world and like just see the traction it's been getting. We did most of that work in like 2020, end of 2020. Some of it was in 2021. So from my perspective, it's like not as exciting as it used to be, just because this is like 2020 stuff, a little bit of 2021. So I thought I would maybe kind of go through the paper, the figures, talk about high level, how we got them, what led to them, and then also talk about more of like what my research is and how I contributed to this. So I guess what would be easier is to kind of like go through my research a little bit, talk about how I do the machine learning, so then it could make sense for the paper. So I guess I'll start there. I'm just I have a few slides prepared from like a presentation I already gave that I could share with you guys.
So
If I
cool.
can I share my screen?
Yeah, of course. Just give me a sec. You don't have the option for sharing your screen?
It says you need to be enabled to share.
All right, give me a sec. Okay, so I'm going to make you co-host, and that will give you the power to share your screen. You should be able now.
Okay. Okay, let me try now. So I just gave this press. Can y'all see my screen?
Yes,
It
yes,
should be like a PowerPoint slide.
all good, yeah.
Right, so I just gave this talk at the Enzyme Engineering Conference last week, so I'm just going to go through a few of these slides. I'm supposed to show this whenever I give any talks in part of UT, that I actually have started a company around my PhD work. It's me and my co-founder, James Loy. And this is all for the MutcomputeX software stack. I don't want it to be confused with the original MutCompute software stack. So whenever I was doing this work, I was using MutCompute, which is developed by Raghav Shroff. He was the 5th-year grad student in our lab whenever I was a 2nd year doing this work. And the way that the algorithm works is essentially, what we do is we go to a protein, we select an amino acid. So you can see right here in pink, we selected this amino acid. This amino acid that you see here in pink is actually the asparagine at position 233, which you'll see it all throughout the Nature paper. So this is the PETase, this is the residue that we talk about throughout most of the paper. And then what we do is we use that amino acid as a focal point to filter out all local chemistry. So this is all Cartesian distance filtering. So once we know the amino acid of interest, we have a point in space, and now we can measure if atoms are close to it in space, yes or no. And then that's what you see here in the middle, where we filter out the local chemistry surrounding that amino acid, and then we generate a learning task for the machine learning by deleting the amino acid we centered around. And then you can see that over here on the right, the pink amino acid has been deleted, right? And then this is what we call a masked microenvironment. And this is how we do self-supervised machine learning with 3D convolutional neural networks. So this is how we generate our data examples. And then here's how we pass it through a traditional 3D CNN, where we have, I think, 4 convolutional layers. So it goes convolution, convolution, pool, convolution, convolution, pool. Then we flatten out all what comes out of that. Then we pass it to a classification layer. And then the output of that is actually a vector of 20 numbers of how probable each amino acid is at that given microenvironment. So given that we deleted a residue, we now get a probability of how likely each of the 20 amino acids is to fill the cavity that we deleted, given the surrounding chemistry. So the models, we get models that can go anywhere from 70 to 85% accuracy. For the model that we used in that paper, let's see, that model had a 69.1% accuracy. A lot of the new ones can easily get to like 83 to 85% accuracy. But the way that we use it for engineering is by looking at when the model makes a mistake, right? The hypothesis is, did nature make a mistake or did the model make a mistake, right? Well, the model's looked at millions of these, and the model's telling us, given the surrounding chemistry, I believe this should be here, right? And that doesn't mean that that the model's wrong. It just means that maybe nature hasn't evolved or had the selective pressure to actually make those mutations yet, right? Are we essentially using machine learning to look forward in time on the evolutionary scale and see what predictions are allowed given the surrounding chemistry? And then that's kind of how we use it for engineering. So for that paper, I worked closely with my friend Brad, and we built an intuitive interface for this. And if I can just find it for you guys. Here, I'll just go to it really quickly. So and this is available for everybody to go see. You just go to MutcomputeX.com. So this is my website that I built for my PhD work. If you go to visualize and you type in the PEDACE that we were working with, so 6IJ6, here is what the AI thinks whenever we do this mass microenvironments, where we go to every residue in the protein, we delete the residue and then ask the model what it thinks belongs there. And then we color it based on the wild type probability. So for example, if we go and look at the 6IJ6 that we were talking about, I mean, the N233 that I was showing you, that's this residue right here. If we click on it, you can see it's red because the model doesn't like it. And if you look in the top right-hand corner, it tells you it's residue 233 and that the asparagine residue gets less than 1% wild type probability and the lysine gets 97% probability. Does everybody see that on the top right-hand side of their screen? So this is a visualization tool for the AI that we built that helps us really see, like, how do we target this protein and approach an engineering campaign? We get to look for regions that are destabilizing. And this is kind of like how we pick the residues to target and focus on. And this is how we actually made the main figure in the paper. So now that I've kind of given you a machine learning background, I'm going to end my session by briefly going over the paper and telling y'all how it came about. Right, so here's the paper. Can y'all see the paper? No problem. Okay, so right, so here is the MutCompute figure I just showed y'all from my website, and here are the residues that we tend to focus on. Here is the figure we made where we make a masked microenvironment, again passing it through the neural networks. And if you see from the data, you can see all of them where we added this N233K mutation had like the greatest impact. So like on wild type, all of the ones that had it. So here's a double variant with it. Here's a triple variant with it, another variant with it. That was the main best mutation. And that's because it gave us about a 10-degree increase in thermal stability, which was huge. And so what I thought was the coolest part of the paper, which isn't really clear, is how we went from figure 1 to figure 2. So you can see that we were talking, the whole paper is about the fast PEDACE and how we engineered this PEDACE variant from the organism, I think it was like Ideonella sicyensis. And pardon my French, I don't know if I said that correctly. But what was really cool about this is that the reviewers weren't really liking the fact that we were not doing a fair comparison with ICCM. And that is because ICCM was published in Nature in 2020. So if we're trying to publish in Nature, they were like, yo, like you need to do a fair comparison with our previous Nature paper. And if you read the details, ICCM works best apparently at 72 degrees Celsius. But our fast PEDACE melting temp was 68 degrees Celsius. So we weren't even able to get it that hot to even do a fair comparison. And they weren't really appreciating that. So it kind of looked like we were not going to get a Nature paper. So that's whenever I was kind of working with Hong. And I'm going to switch back to my presentation where I was reading these papers, that Nature paper. And then there's this other review that recently came out. Can y'all see this figure? This is what I thought was super cool. So whenever you superimpose the cutinase and the PEDACE and other orthologs and homologs of this enzyme over each other, you can see that like they have, while the PEDACE and the cutinase have very different sequence similarities, so 48% sequence similarity, you can see that they have a conserved calcium binding domain, right? And the residues in this calcium binding domain, even though the proteins are very different, are actually very, very almost identical across all the scaffolds at this region. And it was super interesting because in the cutinase paper, what they did to deal with this calcium dependency was they designed the disulfide bridge. And if you read their paper, they actually lost a 40% or so of activity upon doing this, but gained about 10 degrees in thermal stability. We gained about 9 degrees by putting at the same exact position a lysine. So it turns out that our AI model knew that there was a cation dependency here. And rather than depending on an exogenous calcium ion, it predicted to put a cationic amino acid there, a lysine. So we, instead of forming a disulfide bridge here, we used the AI and it told us to put a lysine here. And now we only got about an 8-9 degree improvement in thermal stability, but got actually a boost in activity because we didn't make the enzyme more rigid, right, with a disulfide bridge. So the protein was still able to maintain a lot of the dynamics needed, but was also more thermal stable. So you can see that the way that we got around doing this fair comparison with the ICCM cutinase was we took the ICCM cutinase, we removed their disulfide bridge design, and then added our AI design, that N233K mutation. And you can see that while the cutinase that was engineered had a TM of like above 100, right? With the lysine there was, I think I dropped it down to like 96 degrees Celsius, which is way above 72. So it was still very stable by removing the disulfide bridge. Hello, am I frozen? Can you also hear me?
Yeah.
And then, so here's the really cool part. So their enzyme, by us removing their design and putting our AI design, we saw a 100% improvement in activity, right? By just making the enzyme less rigid and allowing it to operate better. And then this, I think, was really what won over the reviewers and got our paper accepted into Nature, because at first they were really skeptical about accepting us because they did not think that like we were doing a fair comparison with their previous Nature paper. I guess I should like briefly mention like the other two figures. I already talked about this one. I just went over this one and how this figure came about. And I gave you the background story on why this figure even exists. So what's cool here was this was something that I worked a lot with Hong on. So Hong did all the experimental work, and then some of our other co-authors did a lot of the polymer chemistry work. But the idea that I had with Hong in summer of 2020 was we were reading all these PET-ACE papers, and we were just tired of seeing them all being compared to like, PET films that they made down the hall, or they like sourced from some chemical vendor. And it just wasn't really also the story we wanted to tell, because at the time of us doing this work, the cutinase paper came out, right? It was very demotivating. We were like, man, like we're not going to get like an awesome, dope Nature paper. We'll probably end up getting like a Nature Communication, maybe an ACS catalysis paper, because it was just very like deflating. So what we thought about is, okay, well, like how can we like boost the paper more? And we were trying to take advantage of PET-ACE as being mesophilic and how it can work at 30 and 40 C. So we wanted to demonstrate that not only can it work at like the temperature outside on a hot Texas day, but it can also work on the plastic that's hanging out outside on a hot Texas day, sitting in the grass. So I worked with Hong on sampling plastics from so many different sources and like going to HEB, which is like our local grocery store here in Texas. We went to Walmart and we just sourced all these different plastics and showed that our protein actually can eat consumer PET, right? Not some PET that came from some chemical manufacturer or vendor or made in some polymer chemistry lab that nobody at home can relate to. We were degrading what your celery comes wrapped in, or your cookies come wrapped in, right? Things that actually get thrown away and end up in the trash or outside in nature. So that's what you see here on the top figure, where the bar graph is the time it took for it to degrade a hole punch. So we hole punched out the discs to try to get about the same volume. Clearly different containers from Walmart and HEB had a slightly different thickness. So that's what you see here in I think, yeah, so that's what you see here in the dots. So the dots are the mass of the discs, and then the bar graphs are the time it took to degrade those discs. So they're all the same size, but very slightly in thickness, and they're all from different plastics. And then we scaled up the enzyme reaction to prove that we can actually degrade containers by grabbing some, like a cookie container. I forgot what we grabbed exactly. I think it was like a cookie or a pie container. And we literally just scaled up the same reaction we were doing, made it bigger, and just did a time-lapse video. So this is actually a real video. So if you want to see the video, you can find it on YouTube. And it goes from over 48 hours. This was done at 50C. We don't show the data, but we can also get to 100% degradation at 40C, which is more typical on a hot Texas day. So we can still do, and the enzyme is still active at 30C, but it's just not, it's not at nowhere near as fast, but it's still like, we'll get the job done in under like a month or so. So like a month, it's a lot better than years for plastic degradation, but it's not as sexy as like 2 days. So this is the time-lapse video. You can find it on YouTube. And then this was the reviewers really wanted us to compare it against directly on water bottles because that is the #1 PET pollutant. And while our enzyme is able to slowly eat the water bottles in the regions that have high crystallinity, we definitely perform a lot better where there's low crystallinity. But we also showed if you just pre-treat the water bottles, we can then get 100% degradation in under 2 weeks. So there's still probably work to be done on trying to figure out how to get an enzyme to degrade crystalline PET. I don't think it's like super possible because just of like the mere fact of what crystallinity is, right? Like the polymers are stacked really well together. They have great pi stacking. It's going to be really hard for the enzyme to get in there and like, you know, disentangle and break those chains apart without having some external help, usually from heat, to help break them apart. But nonetheless, the enzyme was able to degrade it, especially if you pre-treat the water bottles. And then we also showed that the enzyme has a small amount of activity to other types of polyesters. So this is kind of an active going project in the lab where I'm combining it with some of the new AI stuff that I have. So like all the MutcomputeX. So the big thing about the AI stuff that I do now is that we can show the AI models, these computer vision algorithms, you can show them non-protein atoms. And that's very important if you wanted to make predictions for substrate specificity engineering, right? If you want to move away from a PET polyester to a different type of polyester, right? Maybe, am I frozen? Hello?
You froze, but the audio is good. So.
Okay. Yeah, so like maybe move away from like, you know, aromatic polyesters like PET, maybe to like other aromatic polyesters. So what I'm working on is moving away from PET to PEF, which is like, that's where the industry is moving away from because PEF can be sourced biologically. You don't need to get, you don't have to source it from oil. But the problem is if we start, and it has better polymer properties, PEF does. But the problem with PEF is there's no enzyme that can degrade it. So we start over from scratch. Or like, yeah, we're able to source it biologically now, but we're just going to start accumulating it in the environment again. So like, how do we move the PETase to be a PEFase? So the new AI models, we can see the ligands, so we can computationally dock the ligands and then make ligand-specific mutagenesis designs to help tweak that substrate specificity and also go to more like aliphatic polyesters. So like the ones you see here on the left. So that's like other projects we have going on, as well as we're looking at other types of plastic eating proteins that don't eat polyesters, but maybe eat like polyethylene, polypropylene, polystyrene, or nylon. And then here's just showing that we were able to take the monomers from the depolymerization and regenerate the plastic, showing a circular economy. And yeah, those are the main figures in the paper. I don't know if there's anything else anybody wants to go through. Here are the AI designs, and you can go see these on MutcomputeX.com as well if you just type in the correct PDB code, which I just showed you how to do. Here's the thermal stability and the expression level, showing that the AI designs tend to improve multiple phenotypes in parallel. You can see here, this one got a boost in thermal stability and a massive increase in protein yield. So the AI is multifunctional. Here's the crystallographic data. So this is something that I'm really proud of, because we were going to submit the paper without a crystal, and I did not want to pass up on the opportunity of having the first AI engineered protein crystallized. So I felt really cool about like being able to contribute to the PDB with like having like the first enzyme engineered with the AI models that we have actually show up on the public databases. I'm pretty much done talking. I think I went over all the main figures. I really don't want to go over on time. I just thought I would give you guys some background knowledge of the AI and some background knowledge of how the paper came to be.
I think it's been a great summary. I mean, I read the paper, I enjoyed, but you just like, yeah, like explained in very simple terms. I mean, with the technicalities, but very, very simply. So now, like before going to the Q&A, Sean, why you don't introduce yourself briefly and just mention how it's going to be the breakout room session activity, and then we'll come back to the Q&A. Daniel, would you just spend the last 15 minutes of the
Yeah, hi everybody. My name is Sean Moshasha. I'm a community member in ValleyDAO for the last couple of months, and also a participant in Race Against the Clock this past year. So I'm the living testament of the joining of the groups, and I can say it's a good group to be part of. Daniel, thank you very much for the talk. It was super interesting, especially you touched at the very end on something that I was super curious about, which was the crystallization.
Mm-hmm.
And I thought that was super cool. But anyways, yeah, we're about to go into breakout rooms. It's going to be about 10 minutes. And we're going to have groups of 5 people. And we're just going to discuss a couple questions. How do you like the article? Ivan actually submitted a couple questions to the chat, so you can check those out and discuss for a couple minutes. And what we want to see at the end is for your groups to kind of come to a consensus of 1 to 2 questions that you can then come back and ask Daniel. With that being said, we're gonna jump into breakout rooms, and at the end.
So real quick, I shared my website visualization of the PEDACE that we use to engineer the protein on the chat. If anybody wants to like view it and see what the AI is doing while maybe like coming up with questions.
Awesome.
Yeah, that's cool.
Perfect. So let's jump into the breakout rooms. Let's remember that here's like a friendly environment, free of judgment. Like any question, there's no dumb questions here. So yeah, let's just talk a little bit about the paper and this exciting science and come back with better questions for Daniel. See you back in about 10 minutes. All right, so people is coming back to the room. I'm not sure if there is someone left behind, but hopefully not. So I hope you enjoyed the breakout rooms. We are a community, and community is about people. So having these kind of interactions are very, very important. And just like having the confidence of talking with a friend about something that you might not understand fully, I think is very valuable. So Albert, I'm not sure if you want to share some words about this collaborative effort that we're doing before jumping to the Q&A.
Yeah, really, as mentioned before, it is kind of utilizing the collective intelligence to learn. The best way to learn is through discussion with other people, and especially when these are quite complex topics. They're not easy. And combining that with having the author here and also discussing it with your fellow racers and ValleyDAO community members, it can support in our personal learning, and then potentially we can apply that in our work in our startups and our research. But also the discussions are also a segue into networking with the rest of the community. So a person you might be discussing with today might be your future colleague or startup founder. It could be really anything. So really take this opportunity to also get to know each other, and yeah, connect basically.
Cool. So let's get into the exciting part, the Q&A. Let's try to make it like you can put like questions on the chat, but I think it's better if you unmute yourself and you just raise a question directly to Danielle. As we are not too many, let's try to do it like in an organized way where like I'm not sure, we don't have the raise hand feature here, but yeah, anyway, I'm going to.
Yeah, we do. Stephen is racing.
Okay, so let's use that hand to line up in the queue for Q&A. So Stephen, thank you for joining, and please do your questions too.
Hey, Daniel, thanks so much for this paper and for coming on here to talk with us about it. In our breakout room, one of the things that came up was that the ability to explain what a machine learning algorithm is actually doing is really important as we attempt to convey this work to the general public or even to other scientists. And so we kind of fumbled around in trying to explain what we thought to be the meat of what the algorithm was doing. And we noticed that we had a hard time explaining how the I think, as you said, the local chemistry was fed into the algorithm so that it could make decisions on what residue might be beneficial in that site. Can you talk more a little bit about like how that kind of stuff arrives at the algorithm?
So just to be clear, you understand that we go to a residue, we select all the local atoms, and then we delete the residue. So that essentially is just a list of atoms now that made the filter. And you're asking me, how does this list of atoms get turned into a format that you can pass it then to a computer vision algorithm?
Yeah, right.
Okay, yeah, no problem at all. So this is essentially how we do it. Can y'all see my screen? Can y'all see it?
Yeah.
Right, so here's a microenvironment example. So this is from my new stuff that I can do. So here's like a ligand that's this. So here's a residue that we deleted. This is a serine. You can see it here. It's like kind of transparent and fat. And then this is like its local microenvironment. So we could take all these atoms in this local microenvironment and we digitize them into these voxelized grids. So you can see we form a 9-channel voxelized grid. So we put all the carbon atoms, oxygen atoms, nitrogen atoms, right? So for example, you see this yellow dot right down here. This is the sulfur atom, and here it is. There's one sulfur atom in this box. And if you see like this purple and this purple right here, those are the 2 phosphorus atoms. Here are the 2 phosphorus atoms that are blurred into the surrounding voxels, right? Same thing with this orange right here and this orange right here. Those are these 2 fluorine atoms that are blurred there. Right, and you can see this alpha helix here. You can kind of see the nitrogens in the alpha helix backbone following up through the box. So this is how we turn the microenvironment into a voxelated representation. And then this whole 4-dimensional tensor, it's 4 dimensions because we have X, Y, and Z. And then the 4th dimension is the channel. So it's an element or a physical channel. And then that tensor is what gets passed into the algorithm. Does that kind of answer your question?
It does more so. I mean, one reality is that someone who's not an expert here explaining this stuff to someone else who's not familiar, it's always difficult to decide how to explain it. So for instance, if I was to talk about this with some other lay person who said, "Oh, I saw that you went to that journal club," I would talk about digitizing the atoms, and that helps the model make decisions about what could or should go there.
And so you're asking then, okay, how does the model take the digitized information and actually do any chemistry inferences?
Yes, yeah.
So this is how we turn the atoms that we're used to seeing on our computer into like essentially a version that the machine learning can see, and then we just show it millions of these, and then the model starts figuring out what's in the input environment isn't to give me the right answer, because we know the answer. We know what we deleted, right? I know in this situation, I deleted a serine. So then you just tell the model after every time it sees an example, well, this was a serine, and then it can like tweak its weights and its parameters to figure out, okay, well, what can I do better next time so I can predict the serine? Right. And then if you go read the original paper where we got these algorithms from, they did a salient analysis, which is like a type of machine learning technique to go look at what input pixels is the model keying into to make a decision. So here we don't have pixels, we have voxels, which is a 3D version of a pixel, right? And whenever they did that analysis, well, so what they did was they went to every atom in the microenvironment and deleted it one by one. So therefore they can get the importance of each atom for making the prediction decision. And then that's how they were able to determine how important each atom was for predicting the correct amino acid. And then they were able to elucidate that the model is indeed learning hydrogen bonding, pi-pi interactions by looking at how different residues and how different atoms in surrounding chemistry predicted the amino acid that belonged there. So I can point you to that paper to talk more about how you can do a sensitivity analysis and figure out how the model, what chemistry or properties or atoms in the input that the model used to make the decision.
Yeah,
Great, that makes a lot more sense. Thank you.
I think that's a very important point, like the hydrogen bonding and what chemistry favors the catalytic activity of the enzyme. So we have Sam, Andres, and Stan in the queue. Daniel, do you mind if we extend like a little bit more, like 5 or 10 minutes?
Yeah, I'm here as long as y'all
stop.
need me to go. I don't have anything right immediately after.
All right, thank you, because we have a lot of biocurious here.
And then I added the paper that did that analysis to the chat if you want to go follow up on that. It's one of their figures if you scroll to the bottom.
Amazing.
Thank you.
Thank you, Stephen. Sam, why you don't unmute yourself and go with your question?
You say Sam?
Sam? Or how?
Okay, very good. Hello, all. I'm Sam. I'm over here in Minnesota. I was with group 5, and we had 3 questions. I'll just ask one and go to the end of the line. Here's my question. So your study here is really cool. It relies heavily on the use of MutcomputeX. And the thing about MutcomputeX is that it's trained on this large training set, essentially looking at the PDB database and looking at all known published protein structures. And that's a lot of proteins. But the thing about that is that there's a little bit of a selection bias in terms of there's all the proteins that biology has made, and there's the proteins that are studied in the PDB database. So for instance, there's a selection bias for proteins that humans are interested in. There's a selection bias for human proteins because humans care about human biology and whatnot. There's also selection bias for proteins that could be crystallized because there's a lot of proteins that never got published in PDB because no one was able to crystallize it. There's some other biases and things and whatnot. My question is, how do you imagine that might have influenced the training of your software that made this study possible? How is that maybe a good thing, or maybe how could you exploit that? What are your thoughts on that? How do you think your software might be maybe either like led in the right direction or misled based on the training set that's available?
Yeah, so this is a really good question. This is something I spent a lot of time thinking about, right? Because I'm always having to come up with data sets, and the data sets is what guides essentially how good the model can perform. So we do a sequence similarity threshold filtering. So I have new models where I train with a 50% sequence similarity, 60, 70, 80, 90. So that's what allows for me to like remove duplicate structures that are oversampling a particular region of sequence space, right? Because a lot, like you were saying, proteins that people tend to care about, they'll crystallize not one, but also the homologs and the orthologs and a few point mutations that they did to study their project. And before you know it, the same proteins there like 100 times. So like, how do you remove all that? So we do that by sequence similarity threshold cutoffs to like make sure we're not overly sampling a particular region of sequence space. And then the other thing is those biases aren't necessarily as bad as you'd think, because we only use about 20,000 structures. The new models I use about 23 to 30,000. So like I think the 90% sequence similarity data set has like 32,000, I think. But the cool thing about it is all these proteins are proteins that people tend to care about. So they're usually like simple bacterial monomer or like dimer enzymes, or they're like human proteins for like cancer and other types of studies. And those are usually the kind of proteins that people need help engineering, right? Like I'm working on another project recently. It's probably be like the next paper that comes out, is we engineered a serine dehydratase for colon and breast cancer. And we had to engineer the human variant because the bacterial variant was causing an immune response, right? So it's luckily for us, there's a lot of human proteins in our data set that can help give mutations that are like already biasing it towards humans. And we actually saw like a massive improvement in expression and stability on this enzyme from this. So we try to balance it from the sequence space side, I'm currently working on trying to do a similar, like a structural similarity data set where like I like don't filter proteins out based on their sequence similarity, but filter them out based on how similar their 3D structures are to make sure I'm not overly sampling a certain region of structure space. But yeah, I mean, that's essentially how we do it. And we also don't sample every amino acid in each protein because then that would bias the sampling to bigger proteins compared to small ones. So I think we limit it to either 100 or 150 residues per protein. So either half the protein or up to 150.
Cool, perfect.
Mhm.
Thank you, Sam, for that question, and thank you, Daniel, for the elaborate answer. Andres, why you don't unmute yourself and share your question with Daniel?
Yes, thank you. Well, first, thank you, Albert and Ivan, for organizing this journal club. That's really great. And thank you, Daniel, for being here. Well, I just had a question. Have you and your team thought what would be the next steps for this? Are you planning to apply this enzyme to a pilot scale so to see if it plays out the same as it did lab scale, or how do you plan to move on with this technology?
Yeah, so I'm an AI protein engineer, right? So I just kind of stay in my lane a little bit. I just move on to the next protein that needs to get engineered. The university owns the protein. It's up to them to figure out who they license it to, who they partner with to scale it up. That's all really out of my control. So I try not to focus too much on that. If anything, I'm actually helping other groups that have reached out to me that want to further engineer the protein. And I'm like, sure, I'm more than happy to help engineer the protein even further or engineer homologs, orthologs. But I just try to dominate my lane in what I do, right? I see myself more as like an IP generator, right? And while I'm at the university, I don't really think about the commercialization of the IP. I was telling Ivan and them earlier that I'm trying to commercialize the AI itself and help partner people who have scaling and commercial the commercial side of enzymes to help them further generate more IP and get more engineered enzymes into the commercial space.
I think a very good question, Andres. What I mean, just for curiosity, we will love to see how the technology keep progressing and if really these enzymes can go to the real world to degrade all the plastic that is polluting the environment. Yeah, I think like at least for me, like the question is it going to be like a purified enzyme through biomanufacturing, or maybe like people in this group can think on, I don't know, like using microorganisms like to grow with the biomass and the plastic itself.
Yeah. So one thing that we removed from the paper was, and the nature asked us to remove it because this has already been shown before, but we actually engineered a bacterial strain that can eat terephthalic acid and turn it back into primary metabolism molecules. So we engineered a strain where it can secrete the enzyme can then, it continuously secrete the enzyme, so there's always a supply of it, degrading the plastic and then turn the monomers into like essentially sugar or whatever primary metabolite.
That's cool. Yeah, I think that'll be interesting to follow up. All right, so Stan, your turn.
Hi, it's very nice to see you all for after like our conference, and I actually got very excited about this paper, and it's a very interesting way to approach it. But you know, I just a little bit didn't get like how you like, you know, you do like machine learning here, and you use like a lot of inputs, but like I'm not quite certain, and you know, about like how you can do like, you know, prediction of a functionality, and like more than that, you know, the paper involves like these questions as temperature and pH and how this works in your results.
So just trying to understand your question. You don't understand how the model gives us functional predictions?
Yeah, so and how good is this correspondence with temperature and especially pH function?
Wait, so how good are the predictions at those two phenotypes? Like, are you talking about for the PETase or like for the model overall?
poor model overall.
Yeah, so I guess I can just show you if I made this figure like a week ago, so I can just show it to you. Yeah, so there's this database out there called FireProtDB, and it has all these delta delta G and delta TM values for, so you can see right here, for delta TM, we have 1365, and this is spread out over 226 different proteins. And then for delta delta G, we have almost 3,600, and this is again over the same 226 proteins. And you can see this is the correlation coefficient of the new models I trained. So we have mediumly decent correlation coefficients, and we beat Rosetta and we beat Facebook state-of-the-art language-based models as well. And then, so what does this mean, right? Some correlation coefficient, Pearson, Spearman, that's pretty useless. So here's a better picture of this. So essentially, if the mutation makes the protein 2 degrees more stable, which that's the first row, or it drops the delta delta G by 1 kilocal per mole, so the protein's more stable by at least 1 kilocal, you can see that the 3D ResNets models that I've trained predict those mutations about 46, 44, about 44 to 50% of the time. And you can see Facebook gets it about 66% of the time, 7% of the time, and Rosetta gets it about 36, 32% of the time. Now, if the mutation is actually destabilizing, so it makes the protein thermally stable makes it less thermal stable by 2 degrees, or it lowers the thermal stability, I mean the Gibbs free stability by over 1 kilocal, you can see my model predicts to not mutate those. Right, so don't make this mutation. It's bad for the protein 95 to 97% of the time. So it's pretty good at telling you when a mutation is going to be destabilizing and telling you not to do it. And whenever the mutation can be thermally stabilizing or Gibbs free energy stabilizing, it predicts those about 50% of the time. So this is like the closest analysis I have to answer your question with real data.
I just like one more small question here is like in a university which is like quite close to you, like 1 hour away, Rice, they do like it called direct coupling analysis method.
Okay.
And it's like known for like, if you have like a huge amount of like sequences and you have like how functure correlates over each mutation, you can actually find something like more successful with that. And you use like a huge amount of sequences. Can you try to like how good is correlation between your results and theirs? Have you heard about that?
So what is this method? You're talking about like comparing it versus an MSA?
Called DCA.
What is it called? DCA?
Yeah, directly coupling analysis. It's like when you each mutation, how, if you do a single mutation, you see how it like a function correlates. It's like very simple machine learning model, which doesn't involve a lot of, inputs, but just
as we are running out of time, maybe.
Oh, sorry. Okay, I'll maybe ask it later.
Yeah, you can put it on an email or on Slack.
Yeah, you can just email me or message me on LinkedIn, whatever's easy.
So we are officially out of time, but still we have time for one question. I mean, extra time. Sean, I see that you have your hand raised. Please go on.
Yeah, sure. So yeah, you were mentioning how you at the end you crystallized the engineered protein and you took the structure of it and contributed that back to the PDB. Do you have any plans on incorporating that crystal structure and, more importantly, like the stats and specs of the protein, such as the activity and the temperature stability, pH stability, all that, back into your machine learning model and maybe making that kind of like another one of those layers of voxels?
Yeah, so the model is a self-supervised learn model. So self-supervised learning is a type of unsupervised learning. meaning we don't actually feed in any labeled data, right? We artificially generate the labeled data by deleting a residue. So that's the beautiful thing about the model. It's that I can use it on any protein immediately because I don't need any information from you besides a structure, right? A lot of machine learning models, you have to first generate a data set where they make like 100 mutations and they get phenotype data, and then they can train on that phenotype data to then predict the next time around more phenotype data. We can just start off zero shot prediction, no data needed. That being said, we are working on trying to build new types of models where we can essentially build an encoder that uses structure to come up with like a dense feature representation of a structure, and then train a simpler machine learning model, maybe like an SVM or a simple neural network or whatever, using labeled data, right? So we could feed it in, right? We can, so that's something that we are looking at working on, but I wouldn't say it would feed back into MutCompute because MutCompute is a unsupervised learning model.
Okay. Would you be able to like create your own like little, maybe like sub data sets? So you said you generate maybe like 150 variants and you know, test those out on the lab. Like, would it be of any use to like use those and create its own data set for kind of further improvement on what actually makes the, or predictions of the active site?
Yeah, so 150 variants isn't really much data.
Sure.
Keep in mind, whenever I trained the model, I used like 2.3 million. So just like keeping it into perspective.
Sure, sure, sure.
But no, so we are looking at ways to pass in phenotype data to the model, but it just wouldn't be this model, right? This model isn't trained for that. It would be like a new type of model where we can kind of leverage the feature extraction portion of MutCompute to then train some downstream regressor or classifier that can take in phenotype data for training.
I see. Cool. Well, thank
Great.
you.
So, thank you, Sean, for the question. Thank you, Daniel. So as we are like pushing for diversity and we really champion women in STEM, like I see Inga Van, I'm not sure if I pronounced it correctly, but I see your hand raised, and we might make a short extra spot for your question. So please go ahead.
Thank you. Hi, everybody. I'm based in London. I was also in the breakout room number 6. And our main question was actually a little bit less machine learning based and a little bit more real world based. And it was regarding the fact, like, how do you see this enzyme to actually arrive at the plastic that's going to be thrown in the environment? Because like if you incorporate it into the plastic itself, it's going to start degrading the plastic whilst it's like still in use or even on the shelf potentially. But then if it's gonna end up in the environment, you're gonna have to, I don't know, spray huge amounts of enzyme in the environment. So I'm just curious, how do you see this to work out or be applied in real life?
Yeah, so the university is looking to partner with people who have the bandwidth and the capability to scale the enzyme. So it would really be up to them to make like a pitch to the university and like convince them that they are the best company to move forward with this. But we've engineered an organism that can secrete the enzyme and turn the monomers that it produces into, bring them back into the bioeconomy. So turn them into primary metabolites. So I don't know if they want to go like the organism route. I don't know how like safe or unsafe or risky it is to like just throw organisms out into the environment like that. But yeah, there's just a lot of different avenues to go. The university is definitely wanting to push the enzyme out into industry. They're just trying to make sure like they don't license it to a partner who like really doesn't do anything with it. Like their fear is somebody wants to license it to keep it off the shelf from other people using it, but they themselves don't use it. So we're just trying to figure out the university, that's the university's job on how they're trying to do that. I work closely with that office and the people who are behind the scenes. So that's why I know that they really want to get it out there and get people, you know, figuring out how well can it scale, what can it do, what can it not do. Like they're excited for that. So we're all kind of waiting to see how it plays out. That's kind of all the information I have.
Perfect. Okay, so with that, I think that we come to the end of this session. 10 minutes after, but with a lot of curiosity and a lot of enthusiasm for understanding the science that you're working on, Daniel. So yeah, on my side, I just want to thank everyone for spending this hour with us. This is the first time that we do the journal club, and this is an initiative that was proposed by ValleyDAO. So I will let Albert to give final words about this initiative and how we can continue doing stuff like this. So Albert.
Yeah, first of all, we should definitely keep this up, and we might host another one in 2 to 3 weeks' time. And also we will go out with a follow-on email, and also ask you if you know if there are any other papers that you would recommend and that you would like to see being discussed in these future journal clubs. And then maybe me and Ivan can try to reach out to the researcher, or if you know him personally, you can try to bring them on this call, because it was really valuable to have Daniel here as a awesome supplement to the article. And just the final words on ValleyDAO. So ValleyDAO, it's an open community. It's community owned. That means that anyone can join it and become an active contributor. And there's a lot of things happening. It's also part of a so-called decentralized science, which basically creates the tools required to enable open science. So we'll send out a couple of links on that. You can read on it and join our Discord. Yeah, we'll take it from there, and hope to see you in a future call like this one.
That will be amazing. And everyone here, you know, like ValleyDAO is cool. Check it out. If you can tell your friends about these decentralized autonomous organizations and the ValleyDAO as an example, will be great. Also, if you have friends that are curious to join Race Against the Clock, we are always looking for growing our community. Thank you so much, Daniel. Again, thank you. We are beyond grateful with your time. I saw that you shared your Twitter. Is that the best way to reach out?
That or LinkedIn.
Okay.
My LinkedIn is also AI Proteins. I got the real estate before anybody got there.
Okay. So are you like Daniel or Danny?
I'm Danny,
okay.
usually, but I'm legally Daniel.
All right. So search for Daniel and connect with him if you are interested to follow up on this topic. Thank you so much. Have a wonderful day, and let's connect again. See you.
Thank you.
Bye.